Almost exactly 10 years since BSE was first identified, what do we really know? Now on BBC Two, Horizon begins a disturbing two-part report on the story so far.
Inside this test tube is enough purified BSE to kill 10 million cows. It now seems certain that it can kill humans too. But the scientific community failed to predict that we would be susceptible. They had been baffled by this extraordinary and mysterious agent of disease.
The infectious agent is almost immortal, and it may be impossible to destroy it. These agents are incredibly resistant to almost everything we throw at them. They resist alcohol, they resist boiling, they resist glutaraldehyde.
Ultraviolet radiation, gamma radiation, microwave radiation, potassium permanganate, chlorine dioxide, hydrogen peroxide.
They resist hospital detergents, ionizing radiation, even autoclaving.
Well, we have here, for example, a hot air oven which runs at the same sort of temperatures as your domestic oven at home, which you would reasonably expect to kill all conventional bacteria and viruses. However, if you cook these types of agents, even at prolonged times in this type of oven, you just don't kill them, which means that, of course, if you have meat infected with such agents, you cannot rely on it to kill these agents at all.
We thought it would be interesting to see what would happen if we buried some of these agents. And so I ground up some scrapey brain and mixed it with soil, put it in a flower pot, and closed it in a cage, and used my own garden as a burial site, right here. And what we found was that a good deal of the infectivity remained in the soil after 3 years. We exposed it to temperatures that turned it to ash. And it did not entirely kill the agent. And so every known pathogen of man would have been destroyed by this process, and this was not.
This extraordinary organism was studied long before BSE appeared, because BSE is only a new form of an ancient group of brain diseases called spongiform encephalopathies. All spongiform encephalopathies are caused by the same infectious agent. Many species of animal other than cows are vulnerable to it, but the disease has always baffled scientists. We know that the infectious agent accumulates in the brain over many years and eventually destroys brain cells. The damage is slow but irreparable. It always ends in death. Under the microscope, the brains of all animals suffering from these diseases have the same distinctive holes that turn the brain to sponge. In humans, the disease is called CJD, Creutzfeldt-Jakob disease. We don't know why, but every person on the planet, from Africa to America, rich or poor, vegetarian or carnivore, is at risk of getting CJD in old age. That's been known for decades. But CJD can infect humans in different ways.
We can think of CJD as coming in three forms. The majority of CJD, about 85%, is what we call sporadic CJD. It's present at similar frequency all over the world. The lifetime risk of getting CJD is about 1 in 50,000, wherever you look around the world, whether there's BSE there or not. Then there are these cases that have arisen from tragic medical accidents, where people have been accidentally exposed to something that's contaminated with CJD. For instance, children that were treated for growth deficiencies by giving them growth hormone that was contaminated with CJD. Those are also very rare cases, thankfully, 20 or 30 people probably in the United Kingdom. There are the inherited forms. These run in families, they're extremely rare. None of these three forms of CJD, as far as we're aware, have anything to do with BSE.
But there is one more form of the disease, and it may shed light on our current fears over BSE, because it caused the first, and up till now only, epidemic of spongiform encephalopathy in human history. Early this century, a strange new disease appeared in the tribal highlands of Papua New Guinea. It was called kuru. It means to tremble with fear. Within years, it had wiped out thousands of people in one particular tribe. It was unknown elsewhere in the world. In 1957, scientists went to the highlands to try and find the source of the disease.
They found a population that was dying from this disease, but primarily affecting children of both sexes equally and young adult women. The first symptoms were a little incoordination. They'd stagger a little bit when they walked. Little by little, over a period of months, this became sufficiently severe so that they were unable to walk unaided. In time, the people were unable even to stand and therefore became helpless. Most people with this disease died within 9 months. Because this disease had this peculiar distribution of age and sex, one of the things you think about is endocrine problems. One of the things you think about is genetic problems. You think about toxic causes. You think about malnutrition. All of these were considered, all were studied in detail, and none of them proved out to be correct. The answer turned out, in fact, to be very simple. These people were cannibals. It was clearly being transmitted from person to person by cannibalism.
This tribe ate their dead relatives as a mark of respect during funeral rites.
This was not warfare cannibalism, this was loving cannibalism. In the course of cannibalistic ritual feasting, the body was cut up into parts, and the men reserved the best parts for themselves, and the best parts were muscle. The remaining parts of the body, including brain and pancreas and liver and kidney and intestines, were eaten by the women and the children.
The disease was passed on when women and children ate infected brains. When they developed kuru and died, they were then ritually eaten, and so the epidemic escalated, an epidemic of human spongiform encephalopathy. This tragic cycle was broken when cannibalism on the island ended in the 60s. But the incubation period of this disease is so long that the last victims are still dying today.
The solution to the way kuru was passed from human to human, together with a number of laboratory experiments conducted over the years since, have, without any question, brought persuasive evidence to the idea that infection can be transmitted by feeding.
In 1986, a new form of the disease struck British cattle. It had never been seen in cows before. Brain slides from infected cattle showed the same distinctive holes of all spongiform encephalopathies. And we were eating those cattle brains.
I think the two key questions then revolved around what is the nature of the infectious agent? In precise molecular terms, what is it? And secondly, of course, the burning question became, can BSE transmit to humans? And are we going to see an epidemic of human disease following exposure to BSE?
But scientists working in the field had no answers. So little was known about these diseases and the agent that caused them, that researchers could only guess what the danger to humans might be.
The theoretical possibility is that BSE might be not contagious, but might indeed provoke a disease in humans. Was always there, and every serious scientist has pointed it out that there was a possibility there. However, it was unlikely, and I still maintain that that it was reasonable to assume that it would be very unlikely. The reason is that sheep scrapie. Sheep scrapie has been with us for a long time, and yet there is no linkage of consumption of sheep brain with Creutzfeldt-Jakob's disease. So it appears that there is a very strict species barrier.
Scrapie is the sheep form of spongiform encephalopathy. It exists all over the world. We've had scrapie in Britain for 200 years. It's fatal in sheep, but seems to be harmless to us. Scientists were confident that even eating scrapie-infected brains would not cause disease in humans.
Come on, girls, come on.
But government scientists believed that cows were susceptible to scrapie, and that's what started the BSE epidemic. Cows were fed a cheap protein supplement to increase their milk yield. That supplement included the rendered carcasses of scrapie-infected sheep. The theory is that through this feed, the disease crossed the species barrier from sheep into cows. Infected cows were then rendered down and fed back to more cows, escalating the whole epidemic. It was a kind of cattle cannibalism, chillingly reminiscent of Kuru. For the first 2 years of the epidemic, cows dying of BSE could be slaughtered and sent for human consumption. Their brains ended up in burgers and sausages, meat pies and pâtés. In 1988, the government called on the scientific community for advice. An eminent group of scientists was asked to study all the research on spongiform encephalopathies, most of it about scrapie, and decide whether BSE was a risk to humans. The committee was headed by the zoologist, Professor Richard Southwood.
Back in 1988, it was very difficult to be confident in one's recommendations. The amount of information that we had about spongiform encephalopathies was very small. We knew there was this disease in sheep, fairly widespread, called scrapie, which had been around for 100 to 200 years. It didn't seem to be causing a great deal of problems in the human population. So we based our conclusion that the chance of transmission to humans was remote on the long experience of scrapie. But we emphasised that if that assumption was wrong, the consequences were grave, and therefore steps should be taken to assume that we were wrong.
On this assumption, Professor Southwood advised that cattle dying of BSE should be removed from the human food chain and destroyed. But cattle can be infected with BSE for years without showing any signs of illness. They could be sold and slaughtered looking healthy, but actually incubating the disease. So the only way to safeguard human food entirely would be to remove in the abattoir the most dangerous organs, the brain and spinal cord, from every cattle carcass. The Southwood Committee did not call for this ban.
Bringing in any of these bans, of course, does pose considerable practical problems. One is imposing additional costs on all those concerned. We didn't at that time think the specified offal ban was crucial.
The Southwood Committee decided only to ban offals from baby food. Potentially infected cow brain could still go into adult food.
We were particularly concerned with baby food because we thought there could be a relationship between the size of the body and the size of a dose, and that a much smaller dose, which could be possible for a small amount of brain material getting into the food, could possibly infect a young baby. And so that's why we picked on baby food rather than on adult food.
It really was probably an inappropriate thing to do, to suggest that just the ban of brain tissue should be confined to baby food. We knew from our work in animals, and particularly in laboratory rodents, that the young animal, quite unexpectedly, was actually largely resistant to infection with these agents. And it was actually the older animals of these species which were more susceptible to scrapie infection than the newborn and the neonate.
9 months after the Southwood Committee reported, the government extended the offals ban to all foods, to protect adults as well as babies. But some abattoirs, perhaps believing that BSE was not a risk to humans, did not fully enforce the ban. We now know that infected material was still getting into human food until the end of 1995.
I've often reflected as to whether we wrote our report in too reassuring way, and in some way this dimmed the urgency which was necessary, and obviously one regrets that.
This reassurance was soon echoed by the government and the meat industry, who used it as their scientific guide and justification.
We rely on Professor Southwood and his independent experts who said the theoretical risk of BSE was remote.
We have to base our knowledge of the disease on scrapie.
We are fairly confident that there is no risk to human health.
The public is not at risk from BSE.
You may wonder why we were not more alarmist in our report, but you must remember that at that time we were dealing with a very rare disease in cattle. The human disease is and has remained very rare. Most people hadn't ever heard of it. And if we had been too alarmist, we were in danger of upsetting the whole of the meat industry in Britain and elsewhere in Europe.
But research carried out since the 1970s should have rung alarm bells about the very basis of the Southwood Committee's reassurance, the link with scrapie. This research indicated that scrapie, and all spongiform encephalopathies, can change fundamentally when they cross into a new species.
We've certainly known since the mid-1970s that when scrapie infection is transmitted to and passaged within a new species, that strain characteristics and disease characteristics can change, and that a phenomenon analogous to mutation can occur, and that as a result of that you can generate a strain with new properties, with new characteristics, with altered neuropathology, even with an altered host range.
Let's assume that BSE is the result of rendered, recycled scrapie in the food chain of cattle. There is not a shred of evidence to date that scrapie has ever caused CJD in humans. But scrapie passaged or going into cattle might change the host range of the infection, and therefore we cannot predict whether BSE would be or would not be infectious for humans.
The Southwood Committee did recognise this possibility, even as they wrote their report.
We, of course, had no way of knowing at that time whether the BSE agent was exactly the same as the scrapie agent. Work since has shown that it does actually perform in a slightly different way to the scrapie agent.
The first clue came in 1990, when domestic cats died of BSE. Cats, like humans, had never been susceptible to scrapie.
It was certainly concerning when domestic cats developed a spongiform encephalopathy, and we now know that that is BSE that was acquired by these domestic cats, and now by quite a few wild cats kept in zoos and some other wild animals kept in zoological gardens. And so that really indicated that BSE had a quite different host range than scrapie. It was infecting different species that had not gone down with scrapie in the past. So that was certainly a cause for concern for me. Of course, that doesn't tell us that humans are going to be at any higher risk, but it does tell us that BSE is rather different than scrapie.
Over the years, the list of susceptible species grew longer. Scientists grew more worried. BSE crossed into almost every species exposed to it. This was extremely unusual.
BSE has a lot of very special features which we have never seen before in other types of spongiform encephalopathy. First of all, it's very promiscuous, meaning that it's not choosy in the type of host that it will affect. It can infect basically all species where this has been tried. It will go into mice, it will go into cats, it will go into monkeys. So we are witnessing a very special disease that has a different feature from the other spongiform encephalopathies we knew before, and therefore we have to deal with the problem that it might even infect humans.
People eating potentially BSE infected material were eating a new disease. They weren't eating scrapie anymore. No, the original scrapie that may or may not have triggered off that epidemic, was long gone.
From the beginning, many scientists had been reassured by the link with scrapie and thought it unlikely that BSE would cross the species barrier into humans. But with the scrapie link gone, they faced the reality that we knew little about this new disease and had no clue as to whether it could infect humans.
The species barrier between cows and humans was unknown. You can't just assume that it's going to be an absolute one. Of course, we can't measure it because that would involve injecting humans with BSE, which clearly we wouldn't do.
It's difficult to see how directly we could ever have that information, as we can't do experiments in humans.
But a human experiment has been done. Millions of us ate infected beef. We've been part of a vast experiment for the last 10 years.
With BSE, I think we are looking at the emergence of disease which may affect significant numbers of people. And I think we have to understand in precise molecular terms the nature of this infectious agent and work out how to kill it.
These agents seem to disobey basic rules of biology.
It goes against everything that we've been taught.
The infectious agent is certainly unique in the world of biology.
The first question, what interests almost everybody, is what is the nature of the infectious agent? What is the agent that causes scrapie and BSE, Creutzfeldt-Jakob? And this has been very enigmatic. And this question, what is the nature of the agent, this is clearly the Nobel Prize question. This is what everybody is trying to answer, and there is a lot of competition.
The mystery of this infectious agent made it difficult to predict whether BSE would be a risk to humans. But for decades before BSE arrived, scientists had been searching in vain for the agent of spongiform encephalopathies.
Scientists have been working on this disease for around about 50 years. It's a very difficult disease to work with. And one of the main reasons is that we don't know for sure what the agent is, so we don't have a direct way of identifying it.
Most infections are caused by bacteria or viruses which replicate and spread disease. They can be seen under the microscope, and the way they work is well understood. For years, scientists searched for a virus as the cause of spongiform encephalopathies. But the agent that causes these diseases remained invisible. It could not be seen, nor even detected by any conventional method. And it had an even stranger quality, one that would prove crucial to understanding its true identity. In 1967, Professor Tikva Alper tried to find out what would kill the agent. She took a solution of infected brain tissue and placed it in front of a source of radiation. She bombarded this sample with a dose large enough to destroy any virus or bacterium. In fact, large enough to destroy any known infectious organism. But this lethal radiation had little effect. The mysterious agent was still active.
The results of Dr. Tikva Alper and her colleagues were so remarkable that we tried to repeat them, and I must say we've repeated them twice with absolutely identical experimental results.
The extreme resistance of these agents to all manner of inactivation procedures does tell us that they're quite unlike conventional bacteria and viruses, and that they are some novel type of living organism.
Scientists tried to identify this novel organism by taking a solution of infected brain material and studying it under an electron microscope so powerful that it could magnify objects 100,000 times. Even the smallest virus can be seen using this machine. After years of scanning brain samples, in 1981, scientists finally had their first glimpse of the killer. But these fibrils contain millions of particles of the infectious agent. Even with this powerful electron microscope, it was still impossible to see and identify a tiny individual particle, or to know what these fibrils were made of. The only way to identify the nature of the agent without actually seeing it was to analyze it chemically. The lab in the forefront of purifying infected brain samples was Stan Prusner's in San Francisco. In 1982, there was one scientist there whose job was to discover chemical clues about the agent.
When I came to San Francisco, what we wanted to look for was what was unique about a sample from the diseased brain that was not in the normal brain. What component, what molecule would be in that diseased brain that was never in the normal brain? And so I did some experiments where I looked at proteins and separated these proteins using a gel to separate them by their size, and they give little different bands on the gel. And when I did that, I didn't really expect to find anything right off the bat. And after one of the experiments, I went into the darkroom to develop a film, and I was very surprised when I saw this. And I knew when I saw this gel that this had to be it. There's a fuzzy band that's here in the gel that's in each of the 3 samples from the diseased brain, and it's not in the samples from the normal brain. And this is exactly what we had been looking for. we and everybody else have been looking for some sort of a key, some kind of a molecule that we could say was in the diseased brain and not in the normal brain. And that really forced us to conclude that this protein is part of the agent, in fact, might be the only component of the agent.
In discovering this protein, the lab felt they had finally cornered the elusive infectious agent. The scientists called it the PRP protein, but it became known as a prion. But finding the prion didn't solve the mystery, it deepened it. This key molecule wasn't a fragment of a virus or bacterium, the normal agents of disease. It was simply a protein, and proteins can't be infectious.
1982, I was at a lecture by Stan Prusiner in Australia, and that was the first time I heard him speak, and he was talking about these so-called prion diseases, scrapie in particular, and he proposed that a protein was the infectious agent, and that seemed extremely odd and very novel, and I was not really quite sure whether this could be true.
For a protein to be an infectious agent was a scientific heresy. Apart from water, our bodies are mainly made up of proteins. They're tiny, far smaller than the smallest virus. There are literally thousands of different kinds of protein, and each has a unique role. Some digest our food, some absorb our oxygen, others form neurotransmitters in the brain that allow us to think. But proteins can't cause an infectious disease. That's because they contain no DNA, no nucleic acid, and therefore they cannot replicate. Nothing on the planet replicates without nucleic acid. By dividing and forming a second copy, nucleic acid provides the only mechanism by which all life multiplies. It is the nucleic acid inside every bacterium and virus that allows it to replicate and cause disease. How could this mysterious protein cause disease without any means of copying itself?
To the best of my knowledge, all infectious agents, all pathogens, require the participation of nucleic acid in order to multiply, to replicate. The idea, therefore, that replication could occur without nucleic acid is heresy.
We couldn't figure out how could a protein replicate if it doesn't have a nucleic acid.
This puzzled scientists for years, but tantalizingly an answer had already been worked out 30 years ago. The theory had lain buried in the literature, almost forgotten, because at the time it was written, in 1967, it was dismissed as too outlandish.
I started thinking about several years before, when I had been doing a literature review and looking in some journals in the library at a paper, and I was looking actually up one paper that was written about the nature of the scrapie agent, and when I went to photocopy it, on the second page of that paper, there was another paper, and it was by a mathematician named J.S. Griffith. And in this paper, he talks about self-replication and scrapie, which is one of these diseases, and he outlines 3 ways that a protein alone could replicate and cause disease. Now, he came up with the idea that you would have 2 different forms of the protein. One was abnormal, disease-causing, a rogue protein, if you will, and the other was a normal protein that would be in the cell or in the brain of a normal person. And the essential part of the theory was that the abnormal form of the protein could bind to the normal form and convert it, change its structure to make it an abnormal or a disease-causing protein. And now you can see that if one molecule binds to another molecule and converts it, now you have 2 molecules of abnormal protein, and 2 can bind to 2 more and have 4, and so on until you have thousands and millions of abnormal proteins in the brain, and pretty soon then you have disease. You have so much abnormal protein in the brain that it would cause disease and cause neurons to die. most amazing thing was that the paper was published in 1967. He was a mathematician, wrote this one paper, never published anything again in Scrapie. The insight was really very astounding that someone back then with so little information could see a way that a protein could be an infectious agent.
What Griffiths proposed was an abstract theory, and it was completely ignored by the scientific community. But over the years, clues have been found that indicate he may have had the answer all along. This form of the protein, the prion, is identical down to the last amino acid, but abnormal. This new shape makes it so chemically stable that it resists heat and disinfectants. And because it's simply a protein, without nucleic acid, it can survive huge doses of radiation, because radiation kills by destroying nucleic acid. Once in its abnormal form, the protein seems to have the unique ability to corrupt any healthy PRP proteins that it comes into contact with, and turn them into prions too. This is not replication, it's conversion.
It's a very strange observation that you have these two quite different forms of the same protein with quite different properties. One of them is a killer. If this protein is present in your brain, you're in serious trouble. The other one is a normal constituent of all our brains. And obviously understanding how one converts into the other, and in the longer term how to stop it, is a tremendous puzzle.
Although there are still puzzles, we do know that once converted, prions link together in long, indestructible chains that accumulate in the brain and somehow create the devastating holes that kill. If this theory is confirmed, it will be a revolution in biology. But so far it remains unproven. No one has yet found a convincing explanation for how prions corrupt normal proteins, and so the protein-only hypothesis remains controversial.
The protein-only hypothesis is beautiful.
I very strongly believe in the protein-only hypothesis.
It's intellectually very appealing.
I became largely convinced that, in fact, the protein-only hypothesis is true.
It should not go unmentioned that the protein-only hypothesis, in its current incarnation, does not explain everything.
The idea that it is an infectious protein is an exciting and curious idea, but is one which I do not accept.
It may be that the agent is just a protein, but if it is, it's a quite extraordinary protein, and it behaves in a quite extraordinary way.
This has been an extremely controversial area of science. Prions are novel infectious agents, and there's been a very rapid evolution of ideas. And I think BSE has arrived on the scene in the midst of this scientific controversy. And of course that's added to the complexities of making these sort of risk assessments.
In the late 1980s, while people were eating infected food, scientists were still trying to unravel the mysteries of the infectious agent. The prion controversy had split the scientific community, and this division only made the search for answers more difficult. If the prion hypothesis was right, it was crucial to understand whether the cow prions in BSE could infect humans. To do this meant deciphering the strange rules of the species barrier. All animals make slightly different forms of PRP protein. The more closely related the species, the more similar their PRP is. The key to the species barriers between all these animals lies in how easily the prions from one type of animal can lock onto the proteins of another and turn them into prions too. This is what determines how easily one species can infect another. But it doesn't always depend on how similar their proteins are.
And it really is quite an unpredictable phenomenon. By and large, we tend to think that animals that are more closely related in evolutionary terms are going to be easier to transmit the disease to. But it's not always like that at all. For instance, it can be extremely difficult to transmit the disease between mice and hamsters, which are quite closely related species. It seems to be relatively easy to transmit BSE to mice, which are quite distantly related species. So, there are really some very strange rules operating here. And the only way to find out is to do the experiments. You can't just sit down with a piece of paper and work out how easy it will go from one species to another. It's something you have to determine experimentally.
First of all, scientists tried infecting primates, whose PRP protein was close to humans. But when the marmosets and macaques died of BSE, it still wasn't proof that humans were susceptible. These animals' PRP, though similar, was still too different from ours to be a realistic human model. A closer primate to humans is the chimpanzee. They were never tested.
Had this experiment been done 10 years ago, we would have that answer today. As it is, the experiment, to the best of my knowledge, has still not been done. It is being discussed, and I'm afraid when the answer comes, which will probably be positive, it won't have any significance at all because we'll already know the answer.
In the early 90s, scientists in America decided to try a different approach, to determine the rules of the species barrier on a molecular level, in the test tube.
Evidence from other labs has suggested that the type of PRP protein that an animal makes can determine whether or not that animal is going to be susceptible to infection with the agent from a different species. So what we know is that this protein, this PRP protein, it's composed of a sequence of what are called amino acids, which are linked in a long chain. And that the amino acid sequence of PRP from an animal like a sheep differs from the sequence in an animal such as a cow. And what we're interested in determining is if it's those differences between these two types of PRP that can affect whether or not this species barrier is broken and infection can occur.
Dr. Priola was intrigued by the puzzle of the species barrier between mice and hamsters. Although closely related and with similar PRP proteins, the hamster disease doesn't infect mice, but the mouse disease does infect hamsters. She analysed the amino acid sequences of the mouse and the hamster prion proteins and swapped single amino acids to try to discover how many of them were controlling the species barrier.
Between the mouse PRP and the hamster PRP, there are 16 amino acid differences. And what we were able to do is show that you can control formation of this protein by changing just one amino acid. So one change out of 254 amino acids could be enough to allow the formation of this abnormal protein. If you get the formation of this abnormal protein, you're probably going to get disease. One of the things that this piece of work implies is that very minor changes in the PRP proteins between two different species could really have a dramatic effect on whether or not you get formation of this abnormal protein.
With this discovery, Dr. Priola had begun to decipher in the test tube the complex rules of the species barrier. But although promising, this is painstaking work, and it will be years before it can answer the key question about cows and humans. Meanwhile, a new scientific technique had been developed, one which might at last indicate whether we would be susceptible to BSE. Scientists had found a way to manufacture substitute humans. Transgenic mice.
Transgenic mice are mice which contain a foreign gene that's been inserted into them. And you can make these mice by taking a single-celled embryo, a fertilized egg from a mouse, and injecting in DNA containing another gene. That can be a gene from another mouse, it can be a gene from another species. In this way it's been possible to produce mice that make human prion protein. So they can now be a model, if you like, of a human being. What we can do is inoculate these mice with BSE and see how susceptible they are to BSE, and that may give us some idea about how effective the real species barrier is between cattle and humans.
This work was begun in 1995, but before it could be completed, the human experiment produced its first results. In March 1996, the government announced that they believed the first humans had died of mad cow disease. Science had failed to find the answer in time.
Like everybody else, we were pretty shocked. I mean, everybody was thinking that there might be a theoretical risk of transmission of BSE to humans, but one thing is to say, okay, there may be a theoretical risk, and another thing is to see patients dying from it. So this came as a very sobering experience to us.
I'm on record as being of the opinion that BSE would not represent a risk to the human population. I accept that I may have to revise that view in light of the variant cases of CJD, but of course we still have to wait and see to see if these really are associated with BSE.
If these 10 people have died from BSE, then essentially the entire population of Britain is at risk.
In this warehouse in America, brain samples are stored from victims of all human spongiform encephalopathies, including those from the only epidemic that we've ever known, Kuru in Papua New Guinea. Scientists are now debating whether we in Britain are heading for the second epidemic, and how big that epidemic might be. Tomorrow night, Horizon asks who is at risk and how many may die.
You can see the concluding part of this Horizon BSE special at 8 o'clock tomorrow on BBC Two.